and well-known with motor memories, like learning to ride a bike and chess and all of that. There are well-known interactions with episodic memories that they can facilitate one another. But a lot of us are spending so much time trying to understand just the basic process that there's less energy spent on trying to understand how it interacts with the other systems.
Chiedere come mai si dimentica e perché.
So why do we forget?
So forgetting can be any of the three, you know, if we talk about those three processes, forgetting can be that you never laid it down, that you never encoded it. It can be some failure for the long-term storage process, or it can be, I mean, we really have no evidence that there's true forgetting. It could be that we just can't retrieve it. It could be that it's like a hard drive, you know, that goes bad and you just can't get the information out of it, but you know it's there, the code is still there. So, I mean, we really don't know. It can be any of those, and I would say there's really no evidence that, at least in human memory, that there's true forgetting. It could be that it's just there, we just don't have access to it.
Can you hear me? Yes. Well, I would like to ask, is there any evidence of interference between anti-epileptic drugs for mild cases like absence seizures, short-time absence seizures, and anticoagulation therapy?
To be honest, this is something I'm not qualified to answer, so I don't know. I know about anti-epileptic drugs, but I actually don't know about anticoagulation therapy, so that would not be fair for me to try and answer that question. Sorry.
Ora, se è importante ricordare, è altrettanto importante dimenticare una quantità di informazioni che vengono date ad ogni stimolo. Può essere guardare un film. Se dovessimo ricordare tutto quello che vediamo in un film, la mente arriverebbe ad impazzire. Cosa fa da discriminante fra ciò che dobbiamo ricordare e ciò che deve essere dimenticato?
So who decides what we have to remember and what we have to forget? It's a more, sort of, theological type of...
Well, I guess that, I mean, I'm not sure what level you want me to answer this, sort of metaphysics or theological, or, but I guess you're asking what determines whether we're going to remember or forget something? Is that what you're asking?
No, if I understood well, what he's asking is, do we know anything about the sort of threshold that the brain probably sets to maintain, to preserve some information and not other? Right.
Okay. So the information is stored not just at one synapse, as I've depicted, but at a constellation of synapses. And the experiments that I showed you in the human, where we showed that the information was there, but the subject didn't have access to it, is a good example of such a kind of threshold. So somewhere between the information that's embedded in the hippocampus and the subject uttering the response, let's say, no, I didn't see it, when in fact they really had, are a series of synapses and interconnections that somehow, as a population, give rise to a decision. And there's a lot of work that's being done at more simple levels of decision-making that describe how ensembles of cells must reach some sort of threshold in order to elicit a response. And so it's probably the same kind of process, sort of threshold, that will involve, for example, the novelty of the stimuli, emotional events, and the strength of the stimuli that will determine whether it's actually something that's committed to memory or not. And this is something that's accomplished not just by one cell, but by, brain systems together.
So, yeah, I think there is time for the last question.
Più che una domanda, la mia è un'affermazione o quanto mai una ricerca di condivisione. Mi spiego, sono una psicologa, psicoterapeuta, orientamento psicanalitico e non psicanalitico, a cavallo. Sono molto attenta alle scoperte delle neuroscienze e quindi anche a questa. E quindi questo volevo chiedere. Mi sembra di poter dire che queste scoperte in qualche modo vanno a confermare gli orientamenti psicoanalitici, laddove viene detto che c'è la rimozione. E quindi anche qui mi pare di poter confermare che la memoria viene fortemente influenzata da traumi, quindi che possono essere rimossi. Basta.
So she's saying that she really appreciates the fact that neurobiology and psychoanalysis somehow converge to the same results, because what psychoanalysts found by studying human beings is not very different from what neurobiologists are finding when they study memory formation and memory removal with all the all the processes behind. So it's not a question, but it's a sort of statement, comment. So any other, probably the very last.
Yes, I have two different questions. The first one, which does determine the unidirectional passage of the messenger from the presynaptic to the postsynaptic membrane? Is it a problem of receptors only? And the second question, on the basis of your studies and of your experience, do you think it's possible that a synapse can recognize a different messenger, for example, after a lesion or under a specific stimulus? Thanks.
Okay, so for the sake of brevity, I perhaps glossed over or made too simple the nature of the communication between the presynaptic and the postsynaptic cell. The presynaptic cell releases glutamate, that is for certain, but there's extensive communication in both directions across the synapse. Antonio referred to some work that I did and other people in the field have done showing that there are so-called retrograde messengers, so signals that are generated in the postsynaptic cell that go back to the presynaptic cell. And there are also mechanical molecules that serve to bridge the synaptic cleft and may actually exert mechanical forces in both directions. And now I'm very sorry, but I forgot the second half of your question. Do you remember?
No, actually not.
You don't remember?
Oh yes, she said she was asking if it's possible that a synapse that has a specific phenotype, for example, neurotransmitter phenotype, after a damage could change.
Okay. Yes, so there's a lot of work done on regeneration. So after a lesion, the responsiveness not only of the neuron but of the supporting cells, the glia, and there's clear evidence that after injury, the game is a different game. There are different proteins that are released and different receptors that are expressed. And in addition, for example, in the case of nerve damage, it's clear that there's protein synthesis, like I described in the dendrites, that's happening in the axons. And what people are trying to learn now is what are the best ways of actually turning nerve cells into, or central nerve cells into cells that are like in the peripheral nervous system that can regenerate and reestablish connections. And this is an area that's very active. There's lots of work going on and lots of information about new molecules that can really help the regeneration process.
So we thank Professor Schumann for spending your time to come here and share all our beautiful results and ideas. So thank you again.